I'm Joyce O'Shaughnessy. I am the Celebrating Women's Chair in Breast Cancer Research at Baylor University Medical Center in Dallas. My practice is Texas Oncology, and I'm part of the U.S. Oncology Clinical Trials Network, where I chair our Breast Cancer Research Committee, which is a nationwide network of clinical trialists who all bond together to conduct clinical trials. Triple negative breast cancer does not have the female hormone receptors, the estrogen receptor, the progesterone receptor, and it does not have an overabundance of a gene called HER2, which can cause breast cancers 20% of the time, but triple negative breast cancer does not have that. It's characterized by a very fast cell division rate, highly proliferative, and with an inability to repair its DNA. So it's very genetically unstable. So we treat triple negative breast cancer predominantly with chemotherapy in the curative setting because chemotherapy is the best therapies we have to kill off fast-growing tumor cells. And also, there are certain of our chemotherapy agents, such as doxorubicin, otherwise known as adriamycin, or one of the platinum agents, carboplatin or cisplatin, that are really very good at exploiting the triple negative breast cancer's inability to repair DNA. These agents, the adriamycin and the carboplatin, for example, really chop up the DNA of a fast-growing triple negative breast cancer, and then the triple negative breast cancer can't repair that DNA, and then it dies. But it turns out that we treat triple negative breast cancer differently than other cancers because we cannot target and inhibit the estrogen receptor because it's generally speaking not there. And we cannot target HER2 because the breast cancer does not overexpress the HER2. So we can't use the agent Herceptin, for example. So we really are right now focused on the highly proliferative fast cell division of triple negative with chemotherapy and with its inability to repair the DNA. Having said that, there's a lot of interest in understanding more about the biology, the different types of triple negative breast cancer. It's not something right now we're using in practice every day. But for example, we talked about the main kind that can't repair its DNA and that's very fast growing. That tends to be the kind that responds best to chemotherapy. On the other end of the spectrum, however, about 15% of triple negative breast cancers are, in fact, slower growing. And if you look, which we generally don't because we're not targeting the androgen receptor, but about 15% of the triple negative breast cancers express or have a protein called the androgen receptor like prostate cancer does. And that is a receptor that actually attaches to the male hormone testosterone, because women make a little bit of testosterone. And so the thing is, we don't know, however, that actually inhibiting the androgen receptor on the triple negative breast cancers will lead to more cures for women diagnosed with triple negative breast cancer. We're still doing research studies on that, so we're not targeting that at this moment. It's something and we're not looking for it in the clinic. But triple negative breast cancer is not one entity. There's probably 3 to 4 major subtypes, and it's a fascinating area of research. I'll give you one more example. Recent data, again, very early and just evolving, and not anything we're looking for every day in the clinic, suggests that maybe up to 40% of triple negative breast cancers have some of the HER2 protein and its close cousin, the HER1 protein, otherwise known as EGFR, and that they are talking to each other and they may be driving some of our triple negative breast cancers. Well, that's very interesting because we have a lot of agents that can block HER2 and HER1. So this again is an area of research. But the point is here, the triple negative breast cancer, they all lack the estrogen receptor, the progesterone receptor, and HER2 at any kind of a targetable level. But if you look under the hood, you'll find a lot of variation in the triple negatives. And ultimately, I think we're going to need to know which ones are the true, what we call basal-like triple negative breast cancers, the ones that are very, very fast growing and really respond best to the DNA damaging chemotherapy agents, and which ones are not particularly basal and need other therapeutic strategies. And that is an area of intensive global research.